Okay. Um, yeah, so I just want to start off by uh, thanking some of the other folks in our office that worked on this presentation with me. Scott Peterson, Amber Schmidt, Sergey Chesney, and also Corey Susky, who's a professor on campus at the U of I. I also want to thank our funding sources and a bunch of other people. So this project is funded by the Federal Aid and Sportfish Restoration Program. If you're not familiar with that, um, it's been around for 75 years. It's legislation that was pushed for by people who hunt and fish. And what that does is collect an excise tax on things like fishing equipment and boating equipment. And then those funds are distributed to the states in part based on license sales. So they're dedicated to support sport fish. So habitat improvements, access for sport fishing and boating, and also fisheries research. So really important across the country for fisheries management. And we also work with the Great Lakes Fishery Commission. So that's a binational commission. There are commissioners appointed by the president of the U.S. and also the government of Canada. And they coordinate a lot of the fisheries management activities across jurisdictions, states, tribes, etc., and really have helped fisheries management for a long time. So work with both those groups. I also want to thank the bass tournaments that we worked with to tag our fish. We reached out to several, and there were several that were willing to help. These are the three that kind of worked out to work with. So yeah, say bass anglers, big lake bass anglers, and then the angler's choice. I also want to thank the Waterfront Pub and the East Chicago Marina. Those are places that we tag our fish. So we're glad to be able to have done our work there. And finally, there's a couple of people in this picture, but really there's a whole bunch of people at our office as well as on campus in Champaign that have contributed to this work and pulled together as a team to make this happen. So I really appreciate that. At these kind of meetings, I'd like to start off by talking a little bit about who we are at Lake Michigan Biological Station. So we are part of the Illinois Natural History Survey. If you're not familiar with the Natural History Survey, it's a sort of a unique organization. There's not a lot of similar entities in other states, but we're part of the Prairie Research Institute at the University of Illinois Urbana-Champaign, and it's a whole wide variety of ecological and natural resources research. Lake Michigan Field Station has been around since 1985, and our work really contributes to the science-based management of Lake Michigan and its fisheries. So we do things like monitor sport fish populations and their invertebrate prey resources. We do monitor the sport fishery itself through our creel surveys. We do a lot of reef work, including offshore reef mapping spawning habitat for lake trout, as well as looking at some of these nearshore constructed erosion control structures. As part of the university, we have capacity to have grad students do some really focused research. So we've had recently some people looking, for example, at eel perch predation on round gobies, as well as colonization of some of these rocky structures by invertebrates, along with a whole bunch of other work. And more recently, now we've been working on smallmouth bass movement. So smallmouth bass are really important sport fish, not just in Lake Michigan, but throughout the Great Lakes region. And they're really well studied in places like Lake Erie, Green Bay, Sturgeon Bay area, and the Beaver Island archipelago in northern Lake Michigan. They're also abundant in southern Lake Michigan and the Calumet River system. And the Illinois DNR does a good job of surveying these through some electrofishing at a couple of sites every year. There's still a lot that we don't know, especially related to what they're doing and where they're moving throughout the year. And that's interesting because this is a very altered system in our area. So a lot of constructed break walls, other infrastructure like bridges and piers, some warm water discharges, a lot of shipping traffic. So it's definitely not Beaver Island, right? But smallmouth bass are still doing pretty well. So we want to know what habitat they're using. We also know that there are a lot of bass tournaments, not just in our area, but throughout the Great Lakes. Many of these bass tournaments are using sort of this weigh-in format where fish are caught by anglers across a broad area and then brought to a centralized common location at the end of the day, weighed and then released, and then the anglers with the most weight win the tournament. So sometimes these fish have been caught, miles apart, and then they're all released together in one location in a batch. So going into this, we had a couple of big questions. We wanted to know how bass are using different habitats throughout the year as the seasons progress. And we also wanted to know what happens to those bass after they're released at these tournament weigh-ins. So we do have a dive team at LMBS. So if only we spoke smallmouth bass, we could just go down and ask them, right? But we can't do that, so we're going to try to answer these questions with acoustic telemetry. The telemetry is just a word that talks about gathering data at remote points for monitoring something. So it's used in aeronautics and meteorology, and in fish and wildlife, we're generally talking about tagging animals and tracking movements. So you may be familiar with people using a big antenna like this to track things like deer with collars on them that are producing a radio signal. Radio waves don't travel well through water, so especially deeper water. So we're using acoustic telemetry instead. And that's using sound waves that are really high frequency above the audible range for humans as well as these fish. And so we have these tags that are producing a high frequency signal. And then we have receivers that are listening and detecting those signals from the tags. So we are implanting our tags surgically into fish. Each tag produces a unique signal, so we can identify individual fish. Our tags are lasting about 3 to 3.5 years. Generally, larger tags will last longer. I know that some people studying sturgeon, for example, have tags that are lasting 10 years. But as you shrink your fish and you have to shrink the tags to fit into the fish, you really become limited. So really small fish, you may be able only to follow them around for a couple of weeks. And we are using, rather than following fish around with a hydrophone or something, we're using stationary receivers. And that's really an efficient way to collect a lot of data with relatively little effort. So we can set these receivers out and they're just constantly listening. And every time a fish is within range of the receiver and that ping from the fish is picked up, it will record that data with a timestamp. And then we can swing by every 6 months or so, download the data off the receivers, and then get our data that way. And using arrays of multiple receivers really allows us to look at long-term patterns across a landscape and where fish are moving and hanging out. So this is our array so far. We have 9 receivers in the Calumet River system, including Lake Calumet, and then 9 more receivers in southern Lake Michigan. But there's really a much larger array out there. So we're part of this GLaDOS network. That's the Great Lakes Acoustic Telemetry Observation System. And there's actually, together with several other researchers, there's hundreds of these receivers across the Great Lakes. You can see that there's so far not a whole lot in southern Lake Michigan yet, but there are plans to expand some of these receivers into our part of the lake. And there's a couple other groups in southern Lake Michigan that besides us that have a few receivers out as well. And this GLaDOS program is administered by the Great Lakes Fisheries Commission. So getting back to our big questions, to answer them, we tagged 50 fish between August and October of last year. So some of these fish have only had their tags for just a little while. So it's really hard to kind of answer the first question. They haven't been out there that long to look at how they're moving through the seasons. So we're going to focus today mostly on the second question, which is talking about how bass are dispersing after these tournaments. So we visited and tagged fish at 5 different tournaments in 2024, 3 at East Chicago Marina on Lake Michigan in Indiana, and 2 at Waterfront Pub on Calumet River. So this is a sort of a typical tournament setup. You can see the anglers lined up over here, and they're weighing the fish at this table. Tournament organizers are keeping track of the weight for each angler. And then I'm kind of lurking over here to the right, hoping to intercept people, see if they'll give us their bass. And then we would do the tagging underneath the tent there. And all of our fish were then subsequently released on site. So we've, again, visited 5 tournaments, tagged between 9 and 13 fish each day. I do want to point out the second tournament here, especially we were at Calumet River, we were limited by the number of smallmouth bass that we had. So there's a lot of largemouth mixed in with those river tournaments, but we were only interested in tagging smallmouth. So we ended up tagging 28 Lake Michigan fish and releasing them back into East Chicago Marina, and 22 Calumet River fish and releasing them at a waterfront pub. So Lake Michigan fish averaged about 17.5 inches and 3.1 pounds. We did have one fish over 21 inches, almost 5 pounds. Calumet River fish were slightly smaller on average, but there were still some respectable fish in the mix. So before we talk about how those fish are moving around, I'll talk a little bit about some of our limitations from our receiver data so far. So I've crossed out 2 of our receivers here. Those are receivers we were not able to get to before winter set in, and we haven't had our boat on the big lake yet this spring. And then we had 15 receivers that were last visited and downloaded in mid-November of last year. And then just a few receivers that we have been able to get some data from last week, actually. So most of what we're going to talk about today is just through mid-November, so relatively short time frame. All right, so this is some movement results for one of our fish. This is an abacus plot. So this is one fish, Jake, our first fish that we tagged. And each row here is a different receiver at a different location. So L7 here is East Chicago Marina. L8 is to the north along Inland Steel. And then L9 is to the east toward Gary. So this fish was released on August 17th, hung out in East Chicago for a couple days, then left, initially went to the north and then back east towards Gary. And you can see it kind of looks like it was moving around a lot between these 2 receivers, but really we are able to detect fish up to roughly a kilometer, maybe a little more away. So it was likely hanging out somewhere between those 2 receivers for much of that time. So we can compare that to the movement patterns of some of our other fish. So Elwood is our second fish that we tagged. So again, released into East Chicago Marina, moved out after a couple days to L9, which is again over by Gary, and then came back to the marina, moved back and forth a couple times, and then was actually gone by September 2nd. So was off our map. We had another fish named Frank Lloyd Wright. So this fish stuck around for quite a while in the marina, but did eventually leave. So that gave us some hope that it was alive and swimming. But it was connected on those 2 receivers just outside the marina in the open lake, and then it returned to the marina. Really liked hanging out there. It was still there as of mid-November. And a 4th fish that did something completely different, Jane Adams. So this fish we released at one of our later tagging dates in October, and that moved initially to the north or to the east, and then to the north, and actually found its way over to the Calumet Harbor area, so L3. And then came all the way back to East Chicago and was in the marina for a few days, and then went back west again and was detected on L1, L3, and L4, which are all in Calumet Harbor. So these are 4 fish, and the point of this is that in these kind of studies, you want to look at a lot of fish because 1 or 2 fish isn't going to really tell you a lot about what the population as a whole is doing. Each fish is doing something slightly different, and that's why we released 28 fish at East Chicago. So this is a little bit different at this spot now for one site. This is at the East Chicago Marina on the receiver there. And now each row is a different fish. And you can rest assured that we have names for all of these fish. They don't fit very well in here, so these are just the tag numbers. But you can kind of see our 3 tag events, right? So, bam, we had 8, a bunch of fish released on August 17th. Bam, September 14th, another batch of fish. They gradually moved out. Bam, October 19th, we had more fish tag, and they trickled out over time. Overall, yeah, I mentioned that already. Overall, our average time for fish to leave East Chicago Marina was about 5.3 days. 1 fish left after 22 minutes. And then Frank here stayed around for 25 days. So we can look at what those fish did then after they left, right? So these are just the Lake Michigan tag fish. The blue circles on here are size relative to the numbers of fish that were detected on each of our receivers. The black points here are receivers that were out there but never detected any of these fish. So none of our Lake Michigan fish went into the river. So at least as of mid-November. We did detect 100% of our released fish on the East Chicago Marina, so all of our tags were working. We'll zoom in now on just where we had detections. So almost 86% of those fish were detected to the east near Gary. 3/4 were detected in the steel breakwall. And then smaller numbers were detected over here near Calumet Harbor. So breakwall doesn't show up on this map, but these 3 sites are within the Calumet Harbor breakwall. And we had just 1 fish detected. This site is just outside the Calumet Harbor breakwall. And again, none of these Lake Michigan tag fish were detected in the river through mid-November. So we can do the same thing for the Calumet River fish. Again, we had 100% detection at the release location. So again, all the tags are working. Average time to leave the waterfront pub location was a little bit longer, 8.6 days. But again, a pretty wide range between fish, 21 hours to 21 days. None of our Calumet River tag fish were detected in Lake Michigan as of November. So we can zoom in again on where we did detect fish. Yeah, I'm not sure if I pointed this out before, but this site here is just on the Lake Michigan side of the T.J. O'Brien Lock and Dam. So that's a potential barrier to fish movement, right? So these fish were released downstream of the dam, and then 82% of those fish were detected on the upstream side of that dam. So most of those fish, as of November, had passed the dam. 45.5% of those fish were detected further up the Calumet River, 2.6% in Lake Calumet, and then 9.1%, that's 2 fish, detected a little less than a mile from the river mouth there. You can see this box is in green. That's 1 receiver that we were able to visit last week, this spring. So one of those fish was there all fall, all winter. And then another one of those fish just showed up in the last couple of weeks. So you can see as we have these tags out there for the next couple of years, we're going to start to see some interesting patterns. Again, none of our Calumet River fish were detected in Lake Michigan. And then this receiver here to the west another one we checked this spring had 0 detections from any of our tagged fish. And that's the same for, we had a few more receivers out of view here, up in the Calseg and the Little Calumet River. And none of those had any smallmouth detections either. So it doesn't seem like smallmouth are moving, to the west. So zooming in on that lock and dam, again, this is that one receiver on the upstream side of the lock and dam, the Lake Michigan side. We can split this up. Fish above the line were tagged on our first tag date, which was September 28th. Fish below the line were tagged a little over a week later on October 6th. So pretty variable. Some fish crossed a lot earlier than others. Even some of the later tagged fish arrived and crossed the dam before the first batch, some of the first batch. And some of those fish apparently really liked hanging out in that area above the lock and dam. So looking ahead, we hope to, with the next 2 to 3 years of tag life, really start to look at the seasonal changes in habitat use. So this is an animation that was put together by some folks at the office, and really sort of as a first glimpse of what that might look like. So you can see August through November, each month we see more and more fish being out there at large, and you start to see different sizes or different numbers of fish showing up in different areas, like Calumet Harbor, for example. So you might be able to imagine, as we have several years worth of these data, we might be able to see whether fish are clustering in certain areas at different times of year. We can also look at presence or absence in a single location. So this is back in East Chicago Marina. We now have some springtime data included here. And what you can notice is this big gap during the winter months. January and February, basically, where all of the fish left. Even Frank, who really likes the marina there, left for at least a few days. And we can overlay the temperature here. So you can see that most of these fish left as water temperatures dropped into the mid 30s in early January. And then as the water warmed up again this spring, fish started to come back. Some of them faster than others. For example, this fish over here just showed up in the last week or two. So those are the kind of things that we hope to continue looking at, and we're really excited to see what happens through the spring as fish potentially start moving around looking for spawning locations. So this year we are going to deploy a few additional receivers, a couple near Chicago, but also we'll be putting some receivers up near Waukegan and Winthrop Harbor. So there are these new breakwalls that were constructed recently off Illinois Beach State Park, big rock breakwalls, and we want to know whether smallmouth bass are using those or not. So we're actually going to be tagging 20 smallmouth in this area. The Waukegan Winthrop Harbor area. And yeah, we look forward to continuing to work with GLaDOS, and with the hope is that we can begin looking at some other questions related to other species, such as lake trout, potentially yellow perch. And speaking of GLaDOS, I wanted to mention what I think is pretty interesting. Our receivers in southern Lake Michigan detected 4 fish that were tagged by other researchers that are part of this GLaDOS network. 3 of those were lake sturgeon, 2 came from Michigan and 1 from Wisconsin. And we did detect 1 largemouth bass that was released in tagged and released in the Chicago River. So if you were here at last year's meeting, you may remember that Purdue University and the Shedd Aquarium has a project looking at largemouth bass in the Chicago River. So, and one of those fish showed up on our receivers. So that's all I've got tonight. I can answer any questions now if there's time. Otherwise, feel free to reach out to me. My email address there. You can visit our website. This QR code also takes you to the website. So